Okay, welcome back to this hastily constructed DNA and genes and genetics topic. This is the third part on gene expression and mutation. So I am assuming that you've watched and internalized and understood all the stuff that was said before regarding what DNA is like, how it gets turned into protein, how the normal process of protein synthesis works at a GCSE level. Okay, now of course things do change. It's very rare and very unusual, but DNA does change. Normally, the replication of DNA and the maintenance of DNA, certainly in humans, is incredibly precise. But mutations, changes in the gene expression and what the genes are like, does occur. So here's the bit of syllabus we're after. Okay, so describing how genetic variants may influence the phenotype, i.e. the what you look like on the outside, in coding DNA by altering the activity of a protein, or in non-coding DNA by altering how genes are expressed. Now, as you can see, there's a lot of higher tiers down there, higher tier, higher tier, higher tier, which of course is where I would hope you're all aiming for the top and aiming to sort of maximise your grade, which is why you're probably watching this video. So, mutations obviously are things that occur continuously. Most do nothing, very few alter the protein, and some that don't alter the actual gene do still have an impact by regulating when the gene gets switched on and off. That's what we're going to cover and we're going to go through. So, first off, when you hear the word mutant and mutation, right, probably some imagery comes to mind, right, a picture there. What we actually mean is this definition: any change in the DNA that leads to a change in the protein. Now, there may well be changes in the DNA that don't change the protein, but in which case, how would you know that they'd ever happened? So it's a little bit like if a tree falls in the forest and no one's around, does it even make a sound? All that kind of philosophy stuff. But really, the mutations that we're talking about, certainly from a GCSE perspective, are the ones that alter the protein. So they're usually tiny. They're usually absolutely tiny, either one base or a few bases that alter protein product. Okay. Usually they don't do anything. Usually they're entirely neutral, right? Or they're bad. They change the shape of a protein and it stops that protein doing its job. Okay, so they're usually neutral lab. Very, very, very rarely you get a mutation that actually makes the protein better at doing something and makes the organism better at adapting to its environment. And that's where evolution comes in. And we've got that in a subsequent video. Okay, so they're very, very good. Never superpowers, unfortunately. Sorry, Hulk, but you're never going to get superpowers. They're usually the mutations that are beneficial, if at all, are tiny, tiny advantages. So, for example, one of the last human mutations was a thing called lactase persistence, which occurred in groups that farm cattle and drink milk, which basically means you can produce the enzyme lactase well into adulthood. So your superpower caused by a mutation is a slightly higher ability to digest cheese. You're not going to be joining the Avengers with that kind of thing anytime soon, I don't think. So, very, very tiny things. So let's go into how they happen, what happens with them. So, DNA's got three broad functions, okay? And it kind of gets to the point where we kind of don't know what a lot of the DNA is for. We're still figuring that out. But the main one we all know about is it, per the last video, is it codes for protein. So that's coding DNA, the actual genes. There are some little bits outside that coding region that actually regulate gene expression, that are on-off switches, okay, that basically say to all the cellular machinery, this is a gene, you want to turn this on, or this is not a gene, go away, don't turn this on. Okay? Now, if you want to know more about gene expression, we don't talk about that hardly at all at GCSE, go look at the A-level videos. So, there's a lot of them bits and bobs in there that we don't know what they're for, probably leftover evolutionary things that maybe used to have a function and now they don't, or viruses shoved them in, right? Ex-viral DNA, but broadly speaking, you've got the stuff that codes, the stuff that regulates the coding stuff, and then the DNA that does nothing. And it would seem that a vast, vast majority of our DNA actually does not a lot. We're learning more about it as research continues, and that it actually has functions in regulating other genes, all this complicated clockwork. But if mutations happen in non-coding bits of DNA, well, likelihood is they're not going to do anything. Okay, so the vast majority of mutations don't ever get noticed. So this is just in diagram form what we mean. Okay, so these red bits here, these are the genes. Okay, so for example, this might be the code spelling out how to make the protein for insulin, right, which is obviously important in diabetics. And then these bits here, these blue bits, is non-coding DNA, might be molecular switches, and they might be telling the cell, this is the bit you want to make into protein, right? This is an insulin gene. And if those were broken, then the cell's machinery wouldn't know to make these bits, in that these were the bits that were supposed to make insulin, right? And as a result, the person would make the insulin and would be diabetic. Okay, so you've got the switchy bits and you've got the codey bits. Any mutation in those could have an effect that you could actually see. So these on-off switches, they're part of this non-coding DNA involved in switching genes on and off, right? Mutation involves variations in them, and that might mean they get switched on more than they should be, or as per the lactase example I gave you, that you're producing an enzyme longer in your life than perhaps you were before, or they're switching an enzyme off, or switching off the production of a particularly important protein, and that can be bad. Okay? So that's our sort of molecular on-off switches bit. We're going to leave that there because there's not a huge amount you need to know about that for GCSE. So as I say, mutation, random change in the sequence of DNA. Right, let's look at that on a molecular level. So here's a structure. Here's a sort of some DNA. As you know, this is going to be read in triplets, and you can probably see where the mutation has popped up already. If you just play a little bit, spot the difference. And it is, of course, there. Okay, we've had one base change into another base. Now, of course, all of these are read in threes, they're read in triplets. So what we've got here, look, this triplet is the same as this one, so that would code for the right amino acid. This one here would code for the wrong amino acid, because we've got the middle letter there, the middle base is not the one it originally was. And then the rest will be fine. So this is caused often by a mistake in copying, right? All the effects of the things that you know about that cause mutations. Often in the media they use as a shorthand is things that cause cancer, right? But please don't believe the Daily Mail on anything, really, especially not on what affects and mutates DNA. Okay? Although they do seem to be obsessed with dividing the world into things that either cause or cure cancer, and I have no idea why. Right? So that mutation then might have been caused by radiation, a carcinogenic compound, that's what that word means. It means cancer causing, or heavy metal ions, like lead and arsenic and all those kind of stuff can cause changes in the DNA. Okay? So what that could do, of course, is it changes the amino acid sequence, so a different polypeptide gets produced. So as we said, in the original, we get something like that, whereas in our new version, we've now got a different amino acid at that second position. Okay, and that could change how the protein folds and change what the protein does. Okay, now that is obviously only affecting one amino acid. Could just as easily miss a base out altogether or insert one. This is where I need you to work through these questions with me, maybe pause the video at various points. So we should be remembering the triplets, that everything works in threes. So first, the DNA bases AAA, TTT, AAA, GGG code for 4 different amino acids. There you've got AAA, TTT, AAA and GGG, right? Now, stick a C in after the first AAA, right? Write the triplets out again, okay? Write the triplets out again and work out what that would do to the triplets coming afterwards. Okay, I'm going to pause the video there to make that to write that out and make that decision. Okay, done that. Right. So of course what that does, this addition, is it shunts all of the other bases down. So now this triplet system is broken, okay? Because the ribosome is still going to come along and read in those triplets, but now it's basically shunted the last base of every triplet onto the first base of the next one. So everything after the mutation is broken, okay? And that kind of mutation is very, very, very damaging to the protein indeed, okay? So in terms of the shape of the protein, different amino acids, okay, the shape will be changed. If it's an enzyme, it won't have its active site, it won't be able to do its job. Okay? So that's what our mutation does. So if you're given the question, describe the events which result in a non-functional protein from a coding DNA mutation, okay, now obviously specifically within the coding DNA here are the marks that you give. 1, the DNA code will be incorrect. 2, so the template will be incorrect. Now those of you who were paying close attention to the last video will know that that template is this molecule RNA, which of course you don't have to know about at GCSE, but that's what it is. And then that goes off out to the ribosome. Number 3, an incorrect amino acid will be added to the protein chain by the ribosome, right? This will affect how the protein functions because the protein will then have the wrong shape, for example, in an active site on an enzyme. But pay attention to what the question's asking. If it's asking about an enzyme, talk about enzymes. If it isn't, don't. Pretty straightforward. Right, so just to put that in diagram form, okay, so here's a little diagram, pretty standard one. So this is 2 chromosomes here that with the gene on them, so pairs of chromosomes, both have got one version each of the gene for this enzyme, whatever this enzyme does. Okay, so here's our enzyme. There's its active site, right? There's its substrate, and they fit perfectly. You all remember lock and key theory, right? Now, let's say somebody inherited or suffered a mutation in one of those chromosomes. So that's a mutation there that changed that DNA, okay? Now, the possible things that could happen, probably the most likely, right? So changed amino acid due to mutation, but it's not really made any difference to the protein, okay? Protein's active site is still the same shape, still works just as well, so no bother at all, okay? The active site shape is that it could, on the other hand, be actually changed so that the substrate does not fit. That enzyme is therefore non-functional, right? That enzyme does not work. So therefore, whatever that enzyme was making out of that substrate will not get made. And a good example of this is albinism, being albino. So making skin and hair and eye pigments basically involves a whole chain of enzymes to take something in your food, the enzyme, the amino acid phenylalanine, and turn that into melanin, the pigments in your hair and eye and your skin. If any of those enzymes are broken, everything after that won't happen. So that's why some people can be albino for all 3, hair, eye and skin. Some people can only be eyes, some people can only be skin, some only hair. Depends on where that mutation that knocked that enzyme out took place. Okay, and then obviously in very rare circumstances, you might just get the active site being changed so a different substrate fits. Now this, if that's a useful thing, would be a positive step forward in terms of evolution. Now obviously it would depend if the new thing, this rectangular thing, was important, right, and needed to be broken down, because if it wasn't, well, it's a pointless adaptation, right? But this is where the environment drives evolution. Okay, so to finish off then, right, we need to answer these questions. So you might want to pause the video here and try and answer these yourselves. So the questions are: 1, if a mutation occurs in coding DNA, what's the most likely outcome? What is the other less likely outcome? What if non-coding DNA is mutated? All right, what happens there? So you want to pause the video there, have a go answering those questions. Done that? Excellent. Right, so moving on. Number 1, they don't alter the protein. Okay, so the most likely common outcome, of course, is the protein does not get changed. The other less likely outcome is that the protein does change, and that can either be bad, if the shape just doesn't do what it's supposed to do anymore, or in very rare circumstances it can be good. Okay, and that drives evolution. More on that in another lesson. And then what if the non-coding DNA is mutated? Well, non-coding DNA will express the genes near them. So if it's one of these switches that gets mutated, your gene might be upregulated, you might have more of that protein being produced, or you might have less. Okay, so that can have a large effect on the phenotype of the organism. And one of the other modern evolutionary quirks of humans, if you like, is in certain ethnic groups whose ancestors purified water through fermentation, making beer and wine and so on and so forth, an overexpression of the enzyme alcohol dehydrogenase is a recent adaptation to the fact that we started fermenting and started producing alcohol, okay? Because those people who could produce more of that enzyme survived a lot longer than those that couldn't. And if that was the only place that you got your water from, well, that was a big advantage. Okay, right, that is mutations. That is a brief run through the things that can happen. If you want to go further, then do step up to AS level and A level material on the channel. Okay, thanks very much. See you again.